Welcome! This is iGEM Team Heidelberg presenting you the basics of our project on non-ribosomal peptide synthesis and the foundational advance this modular system offers for the iGEM community. Let's start! All of us know the usual way of protein synthesis. It starts with the transcription from DNA to mRNA, followed by the translation into a growing peptide chain by the ribosome. The peptide chain then folds up to its functional structure as a protein. In our case, we deal with NRPS, non-ribosomal peptide synthesis. Here, our protein of interest consists of 3 so-called modules. Each module takes up one specific amino acid. In our case, valine, ornithine, and asparagine. Within this NRP synthetase enzyme system, the specific amino acids are taken up in a template-independent manner. The first amino acid, here valine, is transferred from module 1 to module 2, forming a peptide bond to the ornithine attached to the second module. This dipeptide is then transferred to the third and here last module, forming a peptide bond to the asparagine that is attached to its module via a thiol ester bond. To cut a long story short, NRPS is about an assembly line of modules producing a short peptide called NRP, non-ribosomal peptide. Our valine ornithine asparagine peptide is such an NRP. But what is so special about NRPS? The ribosome has 22 amino acids to work with. NRPS has many more building blocks to choose from, more than 500 actually. The short peptides are assembled in a template-independent manner because every module has a specificity for one single amino acid. The variety of monomers leads to different peptides with several different functions, such as antibiotics, pigments, or agents for recycling, and many more. Seeing all those special things about NRPS, we had an idea. Imagine, with 3 modules and more than 500 different amino acids, there are 500 to the power of 3 possibilities to combine modules. Or to give you an exact number, more than 125 million different non-ribosomal peptides to be created by fusing only 3 modules. We spent the summer investigating the flexibility of NRPS to introduce this novel system into the iGEM community. We were able to produce synthetic peptides by shuffling different modules and found a simple way of tagging the short peptides with a blue pigment. This new tagging system makes high throughput possible. We standardized the process of creating a desired peptide with our NRPS Designer software, followed by detection with our novel tagging system and the screening of products by functional assays. The exact procedure is described in our 2 RFCs, RFC 99 and RFC 100. So now it's your turn to start working with NRPS. To find more detailed information, please visit our wiki, 2013.igem.org/Team Heidelberg. See you in Boston!